That's one question, and that's a subset of my general question, which is what's in store in the future, and can we bring that?
It's on.
Can we bring that 60 or 70% up to 100% by including things like anatomy?
So the first part of Bob's question was about the anatomy of the cell. Actually, E. coli and human cells have very different anatomies. E. coli has very little internal structure, whereas human cells have lots of different compartments, like the mitochondrion. Now, in our and different reactions take place in these different compartments of the human cell. Now, the human model that we've done so far is an uncompartmentalized model. So we're essentially treating the human cell as just a big bag of molecules, which we know is incorrect, as you're pointing out. One way to look at the compartmentalization is that on the one hand, it is imposing constraints on the reactions. So there may be reactions that can't take place in a real human cell because the molecules involved can never see each other because they're in different compartments, whereas in our model they can see each other. And so reactions might be active in our model that can't really take place. And so there's a case where we might be predicting growth of the cell when it can't really happen. And so that's a piece of future work, is to build a compartmentalized model. That's harder to automatically compartmentalize, to automatically build a compartmentalized model. People have already built compartmentalized models. We have not built one yet. But that's another kind of bioinformatics prediction algorithm, is to predict where the proteins end up in the cell. And so that's some future work that we need to do, is to automatically generate compartmentalized models. Let's see, the second part, and in general you were asking how do we improve the accuracy of the models.
And that would be one, the anatomy would be one example.
The anatomy is one way to improve the accuracy. It turns out another limitation on the accuracy of these models is regulation. In what I've described, we assumed that all the metabolic reactions are available to the cell under all growth conditions. Well, that's not true, because the cell turns on and off different genes at different times. So if the cell's growing with glucose available, with oxygen available, there are actually a lot of reactions that are turned off. And again, your models predict growth when they should not, because they have more reactions available to them than is true. And so a number of people are working on that already. Given knowledge of regulatory events in the cell, can you predict what subset of your reactions are actually available? Which subset are turned on, which subset are turned off? So that's an area of active research. That's something that we need to incorporate into our models as well. But yeah, I want to get these models as accurate as we can. Yes? Oh, I'm sorry. Didn't even see everybody back there.
Is this on? You described a process of basically taking a look at two different sequences and comparing them. And then you did sort of a hand-wavy thing with respect to how you actually measure or score differences with respect to these string sequences. And also, how might you a priori try to find a sequence that's similar to one that you already have in hand?
Right, so scoring of sequence similarity searches is a huge research area in bioinformatics. There have been many, many papers published on that topic. And the basic idea is that, well, if you have a database of sequences that's big enough, and if your sequence is short enough, you're bound to find a match. And so you need to somehow distinguish matches that would occur by chance versus matches that are real biology. And so there's a very elaborate area that's built up, actually partly pioneered by Sam Carlin, who was a Stanford math professor who passed away a couple of years ago, to look at the statistics of this problem. And there's a lot of work that's been done to compute a p-value, a probability value, that the match that you found would have occurred by chance. And so all these searches are accompanied by p-values that let you assess the significance of the match that you found. And they take into account the length of your sequence, the length of the database you're searching against, and the length of the match that you found.
And to my second question about if you have a sequence in hand, trying to find a similar one, do you have mechanisms such as hashing or taking pieces of it and looking for subsequences and doing lookups based off that? I mean, what mechanisms are available to actually make those searches?
Yeah, again, a very active area of bioinformatics research that's been around for a long time, and there's a lot of sophisticated approaches to making those matches fast because lots of global warming gases are being emitted by computers doing those calculations. And so people want to make them happen as fast as possible. And yeah, there's lots of hashing techniques, huge literature on that area.
So one of the things I had a question about was, I assume the natural progression here is to analyze existing organisms, and then as you succeed with that, use these tools to help design and optimize modified organisms. And are you at the point of doing the latter yet, or is that still in the future?
Yeah, I mean, there are already algorithms published where, given the metabolic network of an organism, how can you, for example, knock out genes to optimize its production of something that it already makes? So there's already people working on that.
Most of the existing organisms are pretty well optimized. So maybe the more interesting problem is adding genes to produce additional.
Right, people are working on both. The thing about natural organisms is actually they're more optimized to survive in a range of environments usually. That's one of the interesting things is that the fact that their environmental conditions are changing, the temperature, the nutrients they're subjected to, the pH, the acid stress, it's a pretty big constraint actually to have to survive over a range of conditions. So you can actually optimize an organism. If you can control the conditions it has to grow under, you can actually make it grow under those conditions much more efficiently than the natural generalist.
And relative to that, you mentioned biofuels as a target application. Are you at the point where anyone's currently using these techniques to try to optimize or design biofuel generation?
Yeah. Actually, a couple of biofuels companies have licensed our software. Not exactly sure what they're doing with it, but they paid money for it, so they're.
Which ones?
Probably not allowed to say.
Thanks, Peter, for the nice talk. Two questions. First of all, in your flux balance analysis to correlate the biochemical inputs with the outputs, you mentioned one of the key problems is filling in the holes of the missing reactions or perhaps unknown parameters. Now, I've looked a little bit at neural network theory, where you have this black box that tries to map a set of inputs into some outputs. And so do you think it would be useful to apply neural network methods to try to infer some of the missing pathways? Is there enough data?
That one I'm not sure about. I can't see the connection right now, but I'll have to give that some thought.
Okay, second question, very early on, you mentioned that in defining what a gene is, it's important to often recognize the ribosome binding site. There's been some discussion over the last several years about RNA that really never makes it into a protein, but may nonetheless have some regulatory element. And so there may be some, shall I say, lowercase genes that have these regulatory effects. And are you thinking about trying to incorporate some of that?
Yeah, I mean, actually, our E. coli database does describe these small regulatory RNAs that have been published in the literature. We haven't done any prediction work on them, although that's yet another active area of bioinformatics research is trying to computationally recognize these small regulatory RNAs in genomes. I'm not really up on how well people can do on that, but the biology is so complex and there's so many different kinds of things we want to build predictors for. That's yet another one.
Lots of opportunity.
Could you go back to that one slide that showed all of the E. coli genes, the colored one? The where it had all of the genes and you had
I think you're talking about a
Where-
version of this. Let's see.
Genome Browser.
And if we click on the little genome thing here, is that the one you meant?
Yes, yes. When you did the one that showed all of them, okay. There was one you showed, and it seemed to have a pattern of bands, like a
Okay, well, if
yeah,
we,
those diagonals. What were those?
in this diagram, or if we click on one of the genes in this diagram? Oops.
Wasn't it just one of the diagrams?
Later in the slideshow.
Oh yeah.
You might be talking about,
One of your visualization assists.
This one?
That one.
what are the diagonals?
Yeah.
I think that's probably just a visual effect. Let's see. I don't know. I don't think that's anything significant biologically. I suspect it's just some visual effect that happens to come out at this
Okay.
magnification. But
Thanks.
yeah, that is kind of interesting. But I don't think it's anything significant.
Hi. I wanted to bring up the subject of evolutionary change whilst your models are running, or whilst the organism is. The organism does not exist by itself. It has many, many different neighbors, and one of the known mechanisms is lateral gene transfer, which, as we look into, my understanding is, the more we look, the more we find it's more frequent.
So the question is,
how does lateral gene transfer or similar mechanisms affect your outputs?
well, we're assuming a fixed organism. It's hard enough to modify an organism, to model an organism that's not changing. You're right that organisms do change over time, and in fact, there's a paper published a couple months ago where people found that the actual E. coli strain that a number of people are using has some changes from the one that was sequenced that people hadn't been aware of. Those changes are relatively slow. And yeah, in general, people have found that horizontal gene transfer, exchange of genes between different bacterial species, happens a lot more often than people used to think. I'm not an expert on that area, but now I think most people who think about evolution think of evolution as being a combination of horizontal gene transfer and inheritance of information from the ancestor strain.
Thank you.
So what you talked about in terms of using the genome was just the genes. What about all the rest of the genome, which is now beginning to be understood to actually have some important function? How are you planning to use that, or is that completely different problem?
Yeah, I mean, first of all, that's a lot more relevant there are intergenic regions in both humans and bacteria, for example. They're a lot bigger in humans. I haven't read all these papers that have come out recently, so I'm not quite sure what to make of them, but it probably pertains to the regulation. Our knowledge is always changing, and so we have to figure out how to integrate that information, but I think regulation will be one way that it gets used.
Hi, thanks for the talk. So with this tool, you've done a lot of stuff in terms of building models for sort of intra-species, and I've been sort of interested in this area sort of known as synthetic biology, where you take genes from different organisms and kind of mix and match them. I'm wondering if you've built sort of any tools that let you sort of do interspecies things. So say that you have some compound A, let's say glucose, and then you have some compound Z, say some biofuel. Is there some way to sort of traverse this database that you guys have kind of curated to figure out what the most sort of efficient enzymatic transformations to get from sort of compound A to compound Z would be?
Yeah, in fact, we have a grant from the Department of Energy to do exactly that, and we have a early version of an algorithm that does that. So you can give it a target molecule and a source molecule and ask it to look in Metapsych for all the reactions that can get you from your source to your target, and have it come up with a scoring function for those different paths according to different scoring criteria. So we have an algorithm working now, and we're going to try to contact different scientists to try to apply it to problems that they're working on and get their feedback as to how well it works. I mean, that's another real paradigm in this field, is that we computer scientists can do our best to come up with an algorithm, but it's usually naive, and we need to talk to the biologists to learn from them about how they think about the problem and what, how do they evaluate these different pathways and learn from them and work with real problems. Keep ignoring that microphone, sorry. Okay.
It's a very simple question. I've always been curious, if you run the genome sequence for a particular person, say 2 or 3 times, how similar are they? And how much of a problem is this in your modeling?
Well, I'd say in general the bioinformatics techniques they're inaccurate enough that that level of error is not going to affect them, generally. I hate to say it, but that's the truth. I don't remember the error rate in sequencing these days. It's probably on the order of 1 in a million or 10 million bases or something like that. Sequencing has gotten quite accurate, but I don't remember and there are now different companies that make different instruments, so they're going to have different error rates, but the technology has been both going down in cost. Well, actually, as it's gone down in cost, it's gotten both more accurate, but the accuracy has probably leveled off. But they're actually surprisingly accurate.
Well, if there are no more questions, I'd really like to thank you.
Welcome.